My name is Ayan Williams. I've been interning at Dr. Laura Colgan's lab this summer, and also for most of the summer I had been shadowing Carlos, who was a lab technician there. Just some brief background about Dr. Colgan's lab as well as for myself. At Dr. Colgan's lab, the main focus is on memory, but specifically on uncovering the biological significance of different biorhythms in the mind, specifically in the entorhinal hippocampal cortex or complex, and trying to figure out what the functions and mechanisms of those biorhythms are. And then for myself, I'm an undergraduate student at UH. I'm entering my final year, and by May, I hope to have gotten my degree in psychology. Previously, I went to UT, and I started off as neuroscience, but I realized for my personal dreams and objectives, it would have probably been best for me to just switch to psychology. And so let's see, getting into it. Okay, so what I've been working on, in the beginning of my internship here, I was working with the MATLAB, and so I was presented with a lot of data of cellular activity recordings from tetrodes, and when you record with these tetrodes, I found that there's a lot of opportunity for like background noise to get into the data and like kind of mess up the cellular, or just kind of mess up everything. And so it was my job to go in, cut the cellular activity away from the background noise, highlight the cellular activity, clean it up a little, and present that to other researchers. And I assume they cleaned it up even more, and then they analyzed it and did whatever it was that they had to do with that. And then later, when I got to meet Carlos, we began training with rats, and with the rats, our goal was to get them desensitized to human contact. And so it was over a period of like 2 to 3 weeks, and we dealt with somewhere between 12 to 18 rats. I forget how much exactly, but they come in groups of 3, and our method was we would get a cage of the rats, bring them in to the laboratory room. We would take the lid off halfway, let the rats roam around a little bit, let them get comfortable. We would give them treats. Once they started taking the treats from our hand, we would start to pet them. Once they were okay with us petting them, we would start to hold them. And so this process could take anywhere from 5 to 9 days. Some of the rats were really calm, some of the rats were really anxious, some were just wild and they like to run around and try to escape all the time. So that was cool to see the rats' different personalities. And then after a while, Carlos and I got our own cage of 3 rats, and we were able to do a project with them, which I will talk about in more detail a little bit later. Okay, so for the next 3 slides, I'm going to talk about what I've learned in neuroscience, but specifically about the things that I have had to learn to get up to speed in Dr. Colgan's lab. And not gonna lie, it's a lot of papers. It's 9 of them. For the first 6, I'm going to gloss over them in about a brief sentence of like 1 to 3, and then the last 3 are really important because they pertain more to the kinds of things that Carlos and I were working on. So first paper right there talks about place cells, grid cells, and remapping. Place cells fire off depending on where an animal is kind of in an environment, and then grid cells, they're cells that, well, they form a grid, and this grid serves as a neuronal internal map representation of the environment that the animal is in. And so what researchers have found is that the external environment is extremely intricate, and yet these grids are a little bit more simplified. And in order to keep up with the changing environment that an animal is in, the grid cells and the place cells go through what is called remapping. And remapping can either be global, which means they entirely change their position and their orientation, or partial, which means they just change their firing rate. So that's the first paper. I'm going to try to go over these a little bit more quickly. The second paper was about different biorhythms. It specifically talked about theta waves, gamma waves, and sharp ripple waves. And all of these brain waves have animal behaviors associated with them, but in terms of memory, I believe it was the theta waves that were with consolidation of memory, the sharp wave ripples that deal with formation, and then the gamma waves that deal with retrieval. And then the third paper goes into a little bit more detail about grid cells and how it kind of is that grid cells are able to extract all this intricate information from the external environment and then incorporate it into these more simplified grids within the animal's mind. Okay, next set of three. This first one here talks about place cells and grid cells again, but also head directional cells and border cells. Head directional cells fire off depending on an animal's head orientation at any given moment, and then the border cells fire off. Well, they say border cells, but it's kind of a loose definition. A border can be like a river in the environment, an actual wall. It can be like any kind of like lineal marking that separates kind of one environment from the next. And so it goes into detail about how all those kinds of cells work together. This middle one was really interesting. It was kind of dense for me, but I found it like kind of fascinating what it was talking about, because when I was reading that paper, I was kind of relating it to what I would like to do in my grad school studies. And I'm gonna try and get this correctly. So groups of cells, when they're creating action potentials and firing off, they're creating these biorhythms that kind of oscillate. And then another group of cells is doing the same thing, and when they oscillate and come together, they do this thing called phase locking. Sorry, hold on, let me slow down a little. Okay, and then so phase locking, when they do that, it allows these two groups of cells to communicate and share information. And this is important because it's a beginning of looking at cognitive flexibility and how and why it is that that animals' minds are able to accomplish as much as they do. And then this last paper of the first group is pretty self-explanatory. Previously, or at least what I was taught, is that memory consolidation mostly occurs when we are asleep, but this paper shows that when you're awake and the hippocampus is going through memory replay, that also helps with memory consolidation. Okay, so these are the three important papers that pertain more to what Carlos and I was working on. The first one and the third one are pretty similar. They talk about the kind of same kinds of things, and then the second one is the most kind of important one. So the first paper talks about it talks about an experiment in which researchers went in and they inactivated either rats, their hippocampus, or their caudate nucleus using lidocaine injections. And so the hippocampus is related to place learning, and place learning is memorizing and navigating your way through an environment using external markers and cues in your environment. And then the caudate nucleus is related to response learning, which is relying on like your bodily cues to navigate an environment. So with response learning, you're not consciously thinking about exactly where you're going, you're just using your body to get you to your destination. And then this paper implies that, I believe it implies that different kinds of learning may be dependent on each other. So when they inactivated a rat's hippocampus, the rat did not have a preference for either place learning or response learning. But when they inactivated the rat's caudate nucleus, the rats could not necessarily do response learning, but they could do place learning. And so it implies that in order to master response learning, you first have to master place learning. And then the third paper talks about the same ideas, but in humans. I'm just going to skip that one to save time. And then this second paper, a triple dissociation of memory systems, hippocampus, amygdala, and dorsal striatum. So when me and Carlos did our project, we focused on place learning in the hippocampus, but also this kind of egocentric reward-motivated learning in the dorsal striatum. And the dorsal striatum learning is pretty similar, I believe, to response learning, but it's not necessarily the same thing. I'm going to leave out the amygdala, but basically the gist of the experiments that were conducted here is that when the experimenters tested the rats on their ability to, well, actually, let me back up a little. So the researchers went in and they inactivated either the rat's hippocampus, amygdala, or dorsal striatum, and what they found was that when they inactivated the hippocampus, the rats weren't able to do the tasks associated with that, but they could do the other tasks that relate to the amygdala and the dorsal striatum ways of learning. And it follows through. Just to leave out the amygdala because me and Carlos didn't focus on that, but when it came to the dorsal striatum and the researchers inactivated that brain area, they found that the rats could not do the kind of learning associated with that and could not do the task associated with that learning, but the rats could do the other tasks that were associated with the hippocampus and the amygdala. Okay, and so this paper implies that there are types of learning that are independent from each other, and that for humans, I guess, it shows that if you are lacking in one area of learning, you can kind of work your way around that and use other ways of learning. But of course, this paper was done with rats, so I would like to see something like this done with humans. And okay, so here we go. I'm gonna get into some personal anecdotes a little bit. So what I've learned about science, the first things that come to mind is, you know, you be meticulous, you follow the protocol, you follow all the kind of laws of nature and whatnot. Also, Dr. Mickey gave a class for all of us interns here, and I learned a lot about experimental procedures, avoiding biases, a lot of mathematical, statistical things that my mind kind of doesn't attach to so easily. But what I did find really fascinating were the kinds of meta-analysis subjects that we went over. So there was a couple of papers. There was one about how biological significance does not equate to statistical significance. There was another one which I found kind of funny, and it talked about how the difference between what is not significant and what is significant in and of itself is not statistically significant. And then there was a third paper where a group of pharmacists were performing an experiment, and they used these kinds of conventional methods of statistics, I believe, to analyze their data. But really, the way they did that was, it was wrong, and it made their data kind of invalid and unreliable. And they, it would have been better if they were more flexible and used other methods in order to conduct their analysis. And so it made me think a lot, because I love subjects like those, kind of subversing what you think about a subject. And it made me think about how, you know, the natural world around us is really flexible, and we often think about science being this, I'm talking way too fast.
You're doing great, don't worry.
Thank you. I have to cover so much. Okay. So we often think about science being this kind of streamlined, very like meticulous, you know, strict kind of realm. Like all of science is just these disciplines that are very strict. I started thinking about how, if the natural world is as flexible and as dynamic as it is, then we should also be that way with our science. And I guess it's changed the way that I see science and statistics, because I can say that I never quite liked the strictness of science, or at least what I thought was like really stern and strict about science. But after reading those papers, it kind of made me think, well, wow, there's lots of room to move around. There's lots of room to actually be flexible and study things in ways that are beneficial for you. And I'm just gonna move on. So, what I've learned about being a professional, first things that come to mind about this is being mindful of others, be respectful, clean up after yourself. Me, myself, I did not interact much with others during my internship. I either worked independently or I worked with Carlos, but I still had to play my role. I had to, you know, wear jeans to lab every day. I had to clean up after the rats. I had to ask and not assume about things and equipment and that sort of stuff. And before coming to this internship, I was a little afraid of professionalism because I've never worked in what I thought to be areas where there are high standards of professionalism. But then I got here and I saw that a lot of the researchers were also like young 20-somethings, you know, just trying to come up in science and figure out things on their own. And that really helped me to relax a little and realize that professionalism is not difficult. It all just boils down to playing your role and respecting others enough so that they can also play their role and y'all can work together. And, okay, so now we're finally getting to the project that Carlos and I worked on. As stated earlier, you know, I read articles, I worked in MATLAB, and I attended Dr. Mickey's lab, but Carlos and I also got to do this little project of our own. And so this is a really crude drawing I made in paint, but please just imagine that the purple lines on the floor, like that's on the floor, and that's tape on the floor, and then the orange circle is an elevated table, and we use the tape on the floor to kind of like as a marker of where we always start with our rats. So the purpose of this project was to see whether or not rats were using place learning or the kind of dorsal striatum reward motivated but also egocentric learning. And so we had a group of 3, and as usual we got them used to our contact and got them desensitized to like us being around. And then we would take the rats, and out of like 10 trials we would like start them at the start place, and I was standing in the middle. So we would start them here, and then they would run around, and when they got here I would feed them a treat. And the goal was for them to stop there for at least 3 seconds and wait for me to feed them. Science doesn't always go as planned. And so the 3 rats, their names were Wanda, Cosmo, and Timmy. And Wanda and Cosmo were not cooperating. Sometimes they would try to fight me and go like the opposite direction. Sometimes they would just stop. I think Cosmo squeaked at me a couple of times, and I was like, whoa, okay. But Timmy, Timmy was I guess the ace rat out of them, and that really made me see like, wow, these rats really have their own personalities. And so the focus was mostly on Timmy, which you know, that says something about, you know, scientists and their biases, but this was just a little project for us to do and for me to work on for this internship, so like by no means is it an actual experiment, but it's a start. So yeah, so we'd start the rats right here, and then they would run over here and I would feed them, and we did that for 7 days. And then on the 8th day, it was test day. And so here it is again. And so on the 8th day, we started the rats over here in this bottom right corner. And what the goal was, it was to see, okay, are the rats going to run over here and kind of use place learning and stop in the same place? Or are they going to use like their bodily cues, and be motivated to get the treat and stop over here. That's kind of the same distance away from where they used to stop before. And the result was, okay, well, of course, Cosmo and Wanda were not, they weren't getting it necessarily. It sometimes seemed like they were, but honestly, they didn't. But Timmy, what we saw was that Timmy stopped here, and he stopped there consistently. Before in the trials, he stopped there at least 3 seconds, but on this test day, he stopped there and he would not move. Sometimes he ran like a little bit like further out, but he never like went over there. And it was really like fascinating because he would stop there and then like kind of turn towards me and wait. And then, of course, I couldn't give him the treat, but I was like, oh my God, I'm actually getting results. And so that was really cool. There's a lot of things I wish we could have controlled for, because there was a lot of sensory stimulus on the side of the room that was over here. And the rats would, during the training days, the rats would like run over here, and then they would just be too busy like sniffing around and trying to like see things and figure things out. But you know, with Timmy, like eventually he got it and he started just like running around and like in this direction, and not getting so distracted over here, but learning to like wait over here to get his treat. And so, yeah, results was Timmy used place learning in order to figure out where to stop on the track during our test day. Okay, so, oh yeah, here's some data. Okay, so yeah, there's nothing, and then Timmy, he starts to learn on the 5th day, and then by the test day, he's doing 10 out of 10 trials. Yeah, and then the other colors were Cosmo and Wanda, who didn't get it. And then further data, Okay, yeah, so we're wrapping up now, wow. So what was of most value to me? Definitely experiencing lab life, being here in the moment, getting to see what it's like to be a researcher, and if I wish to continue on this path, talking to other young researchers, figuring out where they are in life and trying to figure out where I am and where I'm trying to go. And then also seeing where improvement is needed in myself, especially when it comes to reading scientific articles. I definitely need more practice with that. Yeah, and then lastly, how the internship has impacted me and my future plans. I'm definitely more confident. I definitely feel like I do want to go to grad school. I was always kind of on the fence about if I was going and where I was going to go and like when I was going to go, but now I'm starting to get an idea about what I actually want to do. And then also when I was reading like the papers that Dr. Kogan gave me, I was relating that to the kinds of things I want to do in grad school. As we all know, I'm really interested in like music cognition, and when I was reading about the biorhythms within the brain, that got me thinking about how like music rhythms and like dance rhythms could be related to that. And so that was all really exciting. And then some good news, I applied for a research position at UH, and can't say I've gotten it, but I do have an interview set up, and I have an opportunity to go to Syracuse to check out some graduate schools there. So really exciting stuff. So yeah, I just want to thank you all. Thank you, Dr. Kogan. Thank you, Dr. Nikki. Thank you, Dr. Judy, for letting me come here and experience being here. And thank you. That concludes my presentation.